Hello, Eve. I'm wishing you everyone a very warm welcome to New Phytologist Now, the webinar series that is supported by the New Phytologist Foundation. This is the third in a series that we are supporting of the Tansley Medal winners, and this is the final one for 2021. But please stay tuned. In 2022, we will have more. The plan is to have more webinars and more series associated with New Phytologist Now. My name is Amy Austin. I'm a professor at the University of Buenos Aires and also an editor at New Phytologist, and I have the privilege of hosting this event. And I wanted to just tell you a tiny bit about New Phytologist Foundation, which is a not-for-profit independent foundation based in the UK, and does lots of neat things promoting plant science. It supports two journals, New Phytologist, but also Plants, People, and Planet, and also supports things like this webinar series and finally, the Tansley Medal, which you will have some information about in the chat, which is open to early career scientists who are seeking to promote their visibility and win a cash award, and also publish in the New Phytologist Journal. And so that's the deadline for that is January 15th. So please do be aware of that. So I wanted to introduce, now that my job is mostly over, who's going to actually be running this event. One is the moderator, who is Dr. Francis Martin, who is a research scientist emeritus, research director emeritus at INRAE in Nancy, France, and has been with New Phytologist for a very long time. Since 1991, he's been an editor, and he currently is serving as the director of as section head of the interactions section. And Francis is sort of the perfect person to moderate this because he loves all things mushrooms, especially ones that he can eat, and also the interactions between plants and microbial symbiosis. And that gets me to the final point of introducing our speaker for today, Anna Burkhardt. And she was the winner of the 2018 Tansley Medal. And she is going to talk to us about the evolution of Rhizobium legume symbiosis. And Liana currently is an assistant professor at Penn State University and in the Department of Plant Science. And so without further ado, I'm going to pass it over to Liana for her talk. So we're really looking forward to it.
So it is fantastic to be here today. I'm really excited to be here to talk about my work, thinking about how we can measure the symbiosis of rhizobial bacteria inside and out of leguminous plants. I wanted to start this by saying the extent to which this journey and this academic journey to get where I am now at Penn State University is really thanks to the mentors and institutions that have supported me along the way. And I've listed here my more formal mentors, and I hope you can see their influence and all their ideas and support that has gone into the talk that I'm going to give today. And I would also like to thank the New Phytologist and the Tansley Medal for the opportunity to think about and synthesize all of the ideas that I was kind of flowing around in my head as an early career researcher when I was on the job market a while ago. So if any of you all know my work, you know that I started off thinking about seeds and how the seed life stage and germination processes can influence the life history of plants and flowering time and can have cascading effects on later life stages. And so I really like to think about hidden life stages that might be ignored or not paid attention to because they're hard to measure. And so when I joined Peter Tiffin's lab for my postdoc, I jumped into thinking about this from a slightly different perspective, where I started to think about facultative symbioses. And these are situations where one organism or two organisms are interacting, but sometimes they exist together in symbiosis and sometimes they're existing apart. And these facultative symbioses are ubiquitous, they're important, but often they're fairly poorly understood from the microbial perspective because it's really hard to understand the selective agents that are happening outside of the hosts or in natural environments. And so today I'm going to talk about a very specific one that's near and dear to my heart, which is the legume and rhizobial symbiosis. And so this is a facultative relationship where sometimes these rhizobial bacteria are living in the soil as saprophytes, but they can also live in the rhizosphere of plants and in these really incredibly beautiful, highly derived symbiotic organs called nodules. And inside these nodules, this is where the biological nitrogen fixation takes place. And what? Okay, sorry. Basically where the plant photosynthates from the plant are used by the bacteria to drive the fixation of atmospheric nitrogen into a form that plants and humans and all of us can use. And so this is a really important symbiosis. It's really amazingly cool biologically, but it's also really important ecologically and to agricultural and in both natural and agricultural systems. And so basically this biological nitrogen fixation in terrestrial systems is the major way that we get this atmospheric nitrogen in a form that we can use as plants and humans. It interacts with other cycles like the carbon cycle and the phosphorus cycle. And humans are having a huge effect on this reactive nitrogen cycle. In one sense, we're having a huge effect on this cycle by planting more crops that are legumes. And so the more crops that can form these nitrogen fixing associations with rhizobial bacteria, the more reactive nitrogen is, with the more nitrogen is being put into this reactive nitrogen pool. But even more importantly, we as humans have influenced the cycle by creating the really important Haber-Bosch nitrogen process to create nitrogen fertilizer. And so this has been really important for increasing agricultural yields and feeding the world, but there have also been trade-offs to this innovation because of the fact that this creating nitrogen fertilizers is incredibly energy intensive and energy costly. And if that comes from fossil fuels, then it can create greenhouse gas emissions to create those nitrogen fertilizers. And it can also influence climate change and greenhouse gas emissions on the other side of the spectrum. So once the synthetic fertilizer is put into crop fields, if it's not uptaken into plants fairly quickly, it can basically get converted back towards atmospheric nitrogen. And if it's not converted all the way, it turns into nitrous oxide, which is again a very potent greenhouse gas and is contributing to climate change. So there are various reasons why using biological nitrogen fixation more effectively and leveraging in agricultural systems could help contribute to mitigating climate change and then also to increasing agricultural yields in a changing climate. I'm obviously not the first person to talk about this. We've been leveraging legumes in agriculture for thousands of years and actively managing it for over 100. But there are some persistent barriers that exist to really effectively shifting over from these synthetic nitrogen fertilizers into biological nitrogen fixation. And I just listed a couple of these in my Tanslate Insight, but these are things like the microbes failing to establish in the soil or failing to compete for resources, or over time, them stopping being as good of nitrogen fixers for the crops that they're interacting with. And my kind of thesis and the big thrust of my research program now is to say, well, I think if we understand the foundational principles that are driving selection on these microbes, both in and out of the host, I think that's going to help answer some of these conundrums of how we can better apply these microbes in agricultural systems and to increase biological nitrogen fixation. So when I see rhizobia, I think about them having to go through a microbial triathlon of multiple different environments, including nodule environments, the rhizosphere, and the soil. I also am putting on here culture environments because this is a human-created environment, but one that's very important because it's an environment that we have done most of the characterization of these rhizobial bacteria in. Most of what we know about how they work comes from culture environments. And this is also the environment that microbial inoculants are selected in and grown up in massive quantities before being added into the soil. And so if we're thinking about how the evolutionary trajectories of these facultative symbionts are influenced, we have to think about all the normal things we think about with evolution, like selection and population size, the frequency of exposure to these different environments, forces like drift and mutation. And the sum total of all that will influence these evolutionary trajectories. In my Tansley Insight, I really focused only on the selection component of this process. And I focused in on how in ecologically realistic conditions and in natural environments, these microbes actually, it's relative fitness that actually matters. So it's not, the overall in a single strain experiment, how well each of these strains do, or each of these mutants, it's how they do in competition with other strains. And again, not a novel question. There's been really cool work in the 1980s and 90s on rhizobia competing a wild type and a mutant strain against each other and asking how they do in different environments. But these were primarily restricted to pairwise comparisons because of the difficulty of tracking individual microbes in complex environments. So you might have a fluorescent tag or an antibiotic resistance gene, but it really restricted the scale at which we could ask questions in host and soil and rhizosphere environments. So the big insight in my paper is to say, basically, there are all these new sequencing techniques that are coming online that can be really effectively applied to scale up and ask not about pairwise competition and pairwise fitness, but relative fitness of hundreds, scores, or hundreds of different microbes in all these different sorts of environments, like the soil, the rhizosphere, and host plants. And so I laid out a couple of these different methods in the paper, and they range from methods that allow us to compare hundreds of different mutants in kind of these IN-seq or TN-seq methods, de novo mutations in evolve and resequence methods, methods that allow us to compare scores of strains against each other, which is what I'll be talking about today. And then again, these ideas of synthetic communities where different species are competing against each other. And so I can't get into the details of this given time, but I just want to highlight that a bunch of new papers have come out in the past year or so that really apply these different methods that work across these different scales of genetic variation to ask exactly these questions in really important ecologically realistic environments. So I think moving forward, there's going to be a really exciting time for really thinking about rhizobial adaptation outside of hosts and inside of hosts. I'm going to spend the next little bit focusing on my work and what my collaborators and I work on, which is thinking at the strain level of natural genetic variation among scores of strains of rhizobia. And the reason I really am interested in this strain level variation is, one, it's really hard to study. So classic 16S metrics are not going to tell you who's winning amongst different strains that all share the same 16S sequence. But also from an evolutionary perspective, selection among these lineages is going to be where mutualism evolution and evolutionary processes are taking place within the species level. And so for the rest of the talk, here's what I plan on doing. So I'm going to tell you a little bit about the select and resequence method, and then I'm going to give you some new data and that we've come up with to think about how relative fitness is occurring across different types of host genetic variation. Then I'm going to zoom from nodules. I'm going to zoom over to non-host environments. So I'm going to talk about soils and show you a peek at some new data we have for thinking about selection on these rhizobia in soil environments. And then I'm going to put it all together and say, okay, well, these rhizobia are actually existing in both non-host and host environments. And how might selection in one of these environments be altering selection or adaptation in these other types of environments? Okay, so the system I'm going to use is the legume rhizobia system, or the legume rhizobia system is Medicago Ensifer system. This has been a model system, a genetic model for 20, 30, 40 years, which means we have a lot of tools for working with this. I'm indebted to all the researchers that have created the system and supported it over the years. But from my perspective, what's really cool about this system are two things. One, these Medicago are pretty selective in that they only associate with one of two different lineages of Ensifer meliloti or Cynorhizobium meliloti and Medicae, which allows us to study evolutionary processes within a species. And then there's also a lot of cool life cycle variation in these plants, so ranging from a perennial crop like alfalfa all the way to winter annuals and summer annual species that are naturalized and exist as annuals even within the US, even though the main range of Medicago is within the Mediterranean. So this is the system I'm going to use. And the method we developed when I was a postdoc with Peter Tiffin was to think about, well, you know, we're now getting all this new sequencing data where it's affordable to sequence tons of individual strains of these rhizobia. And what those whole genome sequences do for us is to basically give us a fingerprint where we can then go in and after the fact, figure out which sets of strains won in these different contexts. And so what we do is we have these sequence strains, we grow them up individually in culture, mix them together to form a mixture of scores of strains at approximately equal proportion, and then impose whatever selective environment we want, whether that be with a host or without a host. Re-isolate the rhizobia, extract DNA, and do whole genome pooled sequencing to figure out which strains won and which strains lost. I'd be happy to talk about the details offline later, but we basically use a haplotype reconstruction method called HARP. So here's what this looks like in practice. In practice, here on the left, I'm showing you the initial strain communities. This is about 100 different strains at approximately equal proportion. This is real data from a real experiment. We take that initial community, we pour it on to inoculate plants with it, and then those strains compete with each other to form nodules. And so each nodule might have a different strain that forms an association with the plant, and then we let those nodules grow. Some of the nodules support larger populations of their rhizobia than others. And then after 8 weeks, we pick all the nodules off the plants, pull them together, and then use sequencing to figure out who won. And this is a plant level composite measure. So this is not only who forms nodules, but also which strains are getting the most benefit or have the highest population sizes when they're in nodules. And from a fitness perspective, this composite fitness metric is really an important metric to look at. Okay. So what can we do with this sort of data? So this is our raw frequency data. We can convert that then into a log fold change from initial to final frequencies to get an idea of the overall fitness. This gives us these histograms where each of these is a different strain of rhizobia, and a strain that has zero fitness is a strain that is neither increasing nor decreasing with selection. So any strain on the right here has high fitness, any strain on the left is a strain that's disappearing in the host environment. We can then compare these different environments to each other and basically look and say that non-host environments, actually selection is weaker in non-host environments than in host environments. So we have far fewer strains in these top A17 plants that are increasing in frequency than in soil environments. We can also ask about composition. We could say, okay, different environments are selecting for different strains of rhizobia. And so here's an RDA plot, but like a principal component analysis, each dot's a different replicate, and you can see that there's consistent selection for different strains in these different environments. Which brings me to the big point making here for this talk is that we can actually then compare selection across environments by looking at fitness correlations. So in this particular case, on the top, I'm showing you basically plotting the fitness of a strain in one host and comparing it to the fitness of a strain in another host. And overall, you can see there's a loose, fairly strong correlation. So a strain that does well in one host also will do well in another host. When we look between then host and non-host environments, so if we look at correlations between soil and nodules, you see there's a very, very, very weak correlation. This suggests that rhizobia should be able to optimize and adapt to, or this preliminary data suggested they should be able to adapt independently to soil, to non-host environments and host environments. And then lastly, I want to point out that when we actually compared our soil and culture environments to each other, we see a strong negative correlation, suggesting that any selection that happens to these rhizobia in the lab might actually undermine its adaptation and use in agricultural fields in natural environment, or sorry, in agricultural and inoculant applications. Okay, so we can do a lot of other types of things with this data. I'm not going to get into it today, but we can do things like you do GWAS, so genome-wide association studies, to figure out what genes in these rhizobia are associated with high fitness in different environments. We can use population genetic analysis on those candidate genes to look at histories of selection on those different genes. And we can also think about validating and because these are model systems, we can actually create mutations in novel candidate genes and then test whether our candidates that came out of these different assessments actually influence fitness across these different environments. Okay, so some new data. So I'm just going to show you really quickly some new data on how, so that was basically 4 environments. I showed you soil, host, and culture environments. But over the last couple of years, my collaborators and I have been really scaling up and thinking about lots more types of environments. And I have a couple take-home messages to share from that, those results. So first, what about different host environments? There's a lot of different types of host genetic variation that these rhizobia might experience across environments. They might influence or experience an individual mutation where you have one change in the genome that might influence rhizobial fitness, and or they might experience differences between accessions of Medicago, or they might experience different species of Medicago entirely. And so all of these things are varying across the landscape and influence the selective landscape that rhizobia are experiencing. And we have this awesome collection of individual mutations that folks have been developing in Medicago that have symbiotic effects. I collaborated with, and here's a nice paper by Sonali Roy that highlights these. I focused with my collaborator, Dihana Trujillo, on one of these kind of families of genes that we know influences symbiosis, and basically showed that we can use this method to basically screen for the effect of Medicago genes on the competitive outcomes of rhizobial strains, as well as the classic single strain metrics that we use. So at the individual strain level, we see these positive correlations, but it generally tends to be strongly positive. So we see differences in which strains survive and do well in these different mutants, but they're different from each other. At the accession level, we can scale up to ask, well, I just showed you 2 different accessions of Medicago, but what if we do 202? So Peter and Brendan grew the whole Medicago HapMap panel with competing strains of rhizobia. And I can tell you today that in general, we see positive correlation. So strains that do well in one host do well in other hosts. And it is always positive. Within the species of Medicago, we always see positive fitness correlations across hosts. And it really is not until we get to the species level where we start to see trade-offs, where some strains that do well in one host actually end up doing poorly in another species of host. And so I think that this sort of larger picture idea of how different scales of host genetic variation are altering potentially the evolution and selection on their rhizobial partners kind of could start to lead to a lot more broader questions about the evolution of these rhizobia moving forward. Okay. One more little bit of new data, which is I've talked to you a lot about selection that's happening in a host environment. We've been thinking about moving into these non-host environments. And so we've been doing soil mesocosm data or experiments where we can take these strains, put them into different sterile soil mesocosms, and compete a bunch of strains of rhizobia against each other. And see which ones win when we impose different abiotic stresses like hot and cold and salt and drought. And stay tuned for the details, but I'll just give you a teaser of new data that we're working on publishing, which suggests that in these soil environments, temperature and salt treatments, including cold and heat, are having huge effects on the strains that are surviving and thriving in these competitive soil environments. And I think that this has really interesting, cool implications for connecting back to thinking about legume life cycles, like winter annuals and summer life cycles, because rhizobia are forming associations with hosts at different times of year and are exposed to selection in the soil versus hosts at different times based on these environments that are based on these different host life cycles. This connects nicely back to my dissertation work, thinking about the modulation of life cycles in plants. So just to sum up, basically, we now have started to think more broadly using the select and resequence method to understand how these plants might be evolving, or these rhizobia might be evolving both together in symbiosis with their host as well as apart. In general, we find strong correlations within one of these host or non-host environments, but we're starting to see these clues at this larger level that when we're thinking about host versus non-host environments, that there ought to be very little constraint to these rhizobia optimizing their fitness in these two different life stages and life contexts that they're existing in. And so as a lab, we're hoping to move forward into thinking about taking these questions out into the field. So all of this was in really controlled conditions, but moving forward, we're looking to leverage the strengths at Penn State in an agricultural research station to build collaborations to actually measure these selective processes out in field environments, both within alfalfa variety trials, as whether in these cover crop cocktails experiments where there are multiple legume cover crops interspersed with other crops. And I have a really nice set of folks that have joined me during COVID as I start my faculty position. I'm really proud and excited of the work that they've done in the past few years and what they're going to do moving forward. And with that, I have many, many people to thank. This is a village worth of work, and I'm really grateful to all these people, in particular Peter Tiffin, Brendan Epstein, Katie Heath, Nevin Young, and Mike Sadowski, who have really kind of been part of the team that has moved this whole field of research forward. So with that, I'll take any questions. Thank you very much.
Thank you very much, Liana. We cannot applaud, but I'm sure many people in the audience would like to do so. I'm very pleased to moderate this, to chair this session, and thank you very much for this nice piece of work. And I would like to remember the audience that they can raise questions by using their hand. So raise your hand, or use the Q&A box to do so. And we have quite a few questions already. What do you think of bacteria with the aid of rock dust to increase soil macrofauna and consequently nitrogen fixation with bacteria?
This is a really interesting question. I'm going to say that I haven't looked into this in particular. I have tons of respect for the amazing nitrogen fixation work has been done and is going on in Brazil. I'd love to make it down there and learn from the really cool work you all are doing, thinking about leveraging strains from natural environments. To answer your question more specifically, though, I have not thought about rock dust. I do think that these really important contexts for thinking about trade-offs between multiple greenhouse gases, right? So there's nitrous oxide emissions and there's also methane. And so when we're thinking about developing these inoculants and these solutions, really have to think holistically about multiple processes that are going on, thinking more broadly about all the different functions that these microbes are moving forward in the soil. So I don't think I can answer your question directly, but I'd love to think about it more, and please reach out to me offline and I'd love to talk with you about it more.
Thank you, Liana. There's one question also from Emily, wanting to know, thank you very much for your talk, very interesting. Do you apply selection pressures, stresses individually only? If so, how can the results be interpreted in the context of global change biology, where pressures in natural environments are multiple and complex? So this was one of my questions too.
Right. Yes. You know, as someone who who thinks about and models plant responses to climate change. I absolutely agree that, we really have to start thinking about, and everyone's moving towards thinking about complexes of stressors. And it's not just one thing, it's things in combination. I would say that, we're still in the beginning stages of using these techniques. I have a bioRxiv paper where we tried to look at factorial combinations of nitrogen addition and bacterial density. But I think that we really need to, these are expensive techniques. And so I do think we need to start thinking about combinatorics, but also figuring out which selective pressures and which stressors we want to measure in a thoughtful way, so we don't just explode the size of these experiments really broadly. And so I have a couple grants out actually that I've submitted, actually trying to think about a few of those factorial combinations that I think will be very important, including combinations of drought and temperature stress, which I think are really important for very different reasons. And particularly in Pennsylvania are going to be some of the big shifts that are happening with climate change here. But to answer your question, yes, I think interactive stressors is the way to go. I think we have to be really thoughtful about preemptively asking the right combinations of questions and making sure they're applied, or they actually are relevant in field conditions. That's another reason why my lab is focused on thinking about some of these seasonal cycling out in real environments and seeing if we can take this out to agricultural fields.
Yeah, good. Yeah, but based on your experiment, what would be the main factor, the one impacting the most the fitness in and out of the plants? Cold, heat, drought stress, salt stress. What would be the most effective factor?
We really need to up our mesocosm experiments, but from the mesocosm data I show you, I really think temperature and salt are going to be really important out of plant stressors to think about. I think some of these other contexts like drought and nitrogen addition, one of the things they may alter more than relative fitness is they may alter the number of nodules that's created. And so instead of altering relative selection, they might have an important shift on the relative importance overall of host versus soil selection. And I think that really being thoughtful about whether we expect these factors to influence relative fitness and relative fitness between these strains versus whether we expect it to influence the amount of time these rhizobia spend in and out of these host environments might be to really a useful way to kind of separate out prioritizing some of these stressors for downstream experiments. But I'd hate to I feel like we're just on the tip of the iceberg here, and we've also done things in the lab primarily. So I'd like to hold off thoughts on most important things until we really start getting out into more natural environments. Does that make sense?
Yes, sure. Maybe a last question before moving forwards. How did you identify the nodules were caused by different microbes? By sequencing, or the nodules have different phenotypes?
Yes, so our method is entirely sequencing based. And so we're basically picking all those nodules off, pulling them together, homogenizing them, and then we use differential centrifuging to concentrate the rhizobia. And in particular, we're concentrating not the bacteroids, but the rhizobia that in our particular type of nodule, there's two populations of rhizobia. There's the bacteroids where nitrogen fixation is happening, and then there are rhizobia that live interstitially in the cells. And these are the rhizobia that are going to go back into the soil, and they're going to be the fitness outcome of this interaction that's going to be what evolves and what is the fitness outcome of the interaction. And so it's those we're really focused on in our select and resequence method, is figuring out which strains are going back out to the soil and are at high frequencies inside those nodules. So we are not looking at nodule occupancy. We're looking at this composite fitness metric that's both who's making it into nodules and then who is thriving and dividing inside those nodules. Does that make sense?
Yes, to me, yes. Oh, there's still many questions, but There will be a chat room later on, so I suggest all the people having additional questions to join us later on. And Amy, I'll give you the call, Amy, now.
Yeah, and you can also feel free to reach out to me, but come to the chat. It'll be nice to actually see some of your faces. I don't see anyone's faces right now.
Thanks again.
Okay. So thank you, Francis, for moderating the discussion. And again, I just wanted to thank Liana for a wonderful talk, and also congratulations again for the Tansley Medal of 2018. I hope that it was a nice experience and that it has helped you sort of launch some of your research. So I just wanted to remind everyone that we will continue this series. 2022, we will be relaunching with a different perspective, probably with authors from recently published New Phytologist papers. And just not to forget, the Tansley Medal deadline is January 15th. So all you early career researchers that are out there connected, please do look on the webpage and follow the links that have been put up in the chat to see what the application process actually involves.